Hello, my name is Sophia Colmenares. I am a rising junior studying chemical and biological engineering at Princeton University and a summer bioinformatics research trainee at the Mayan Lab. Today, I will be presenting a project I worked on during the summer titled Benchmarking Methods to Produce L1000 Consensus Signatures. To begin, I will first provide some background on the L1000 assay, an experimental high-throughput transcriptomics technology utilized by the Connectivity Map team at the Broad Institute to measure the response of human cells to thousands of perturbations. The assay consists of a 384-well plate and uses Luminex technology consisting of colored beads and a scanner to measure mRNA expression. Although Luminex technology was designed to detect just 500 bead colors, a novel approach allows the L1000 assay to measure 978 selected genes, also known as the landmark genes. The landmark genes are a reduced representation of the transcriptome and were chosen to maximize the number of inferred genes. By assigning each bead color to 2 genes and hybridizing the beads with cell line-derived mRNA, the Luminex scanner detects both the color and intensity of each bead, which corresponds to the gene and its expression respectively. With these measurements, a computational inference algorithm was applied to infer the expression level of 11,350 additional genes in addition to the 978 landmark genes. The L1000 dataset is generated as a part of the NIH Common Fund Library of Integrated Network-Based Cellular Signatures, or LINCS program. The LINCS L1000 dataset contains gene expression profiles from over 1 million experiments, consisting of chemically or genetically perturbating human cell lines. We process these LINCS L1000 expression profile vectors into signatures. In this context, a signature is the comparison of unperturbated cells to cells treated with a single drug or a genetic perturbation. There are many signatures that correspond to each of the thousands of perturbations. This is because the perturbation was applied to various cell lines. Gene expression was measured at different time points, and for drug and small molecules, the compounds were applied in different concentrations. My goal for this project is to condense these signatures into a singular consensus signature for each drug and gene knockout. This condensed representation is needed for various downstream analyses that I will discuss later. A few words about the CRISPR gene knockouts. LINCS L1000 data. It is the primary genetic perturbation dataset that I worked with. CRISPR-Cas9, otherwise known simply as CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a gene editing technology that can permanently silence a target gene using a single guide RNA, or sgRNA. Unlike other genetic perturbations, such as short hairpin RNAs or shRNAs, CRISPR is designed to silence a gene completely, hence the term knockout used for the CRISPR perturbations versus knockdown, which is used for shRNA. Since CRISPR gene knockouts were profiled for gene expression for the same genes across multiple cell lines, my project was to see which method would produce a consensus signature by combining the information from all the knockouts for each gene. Thus, I will develop methods to generate consensus signatures that represent common gene expression changes after CRISPR gene knockouts. The first step of my project was to pre-process the L1000 CRISPR knockout data, which examined 140,945 unique gene knockout signatures. A total of 5,208 different genes were knocked out across the signatures. Each signature pertained to one of 31 cell lines and varied across 4 different time points after knockout, which includes 10 hours, 96 hours, 192 hours, and 408 hours. This data is in the form of gene expression levels and was transformed into differential gene expression signatures using the characteristic direction method, which computes relative gene expression and differentiates between up and down regulated genes in a signature. The characteristic direction method is a unique differential expression analysis method that was developed by the Mayan Lab. Each CRISPR knockout signature contains 12,327 dimensions, where each dimension represents a gene measured for differential gene expression levels. I primarily analyzed the signature whose gene knockouts were presented in the protein-protein interaction database with the Biological General Repository for Interaction Datasets, or BioGRID. This database will be utilized for benchmarking later on in the project. Once the signatures and the genes were reduced, I then began applying the methods to obtain consensus signatures. 3 approaches were utilized in this project to generate consensus signatures: mean, median, and highest effect. The mean approach involves taking the average of the signatures corresponding to each knockout to form a singular consensus signature. For the median approach, I took the median of the total differential gene expression levels across each signature of a specific knockout gene. Finally, the highest effect approach involves adding up the absolute values of differential gene expression levels across each signature and assigning the signature with the greatest sum as a consensus signature. I also implemented a randomized and shuffled method for the purpose of benchmarking, which will be discussed later in detail. After completing these three processes, I had 5,208 generated consensus signatures from each method. After computing the consensus signatures, I began to analyze the similarity between the knockout genes. To do this, I generated gene-gene similarity matrices with the consensus signatures as input vectors by using Pearson correlation, Spearman correlation, and cosine similarity, respectively. With these formulas shown in the slide, I calculated the respective correlation between each of the knockout genes and the rest of the knockouts to obtain three different distances for each of the three consensus signature generation methods. Here is the visual representation of the similarity matrices outputted by each pipeline. These 5,208 by 5,208 matrices are important to evaluate which genes are highly correlated with one another for the benchmarking process. After obtaining the similarity matrices, I now have completed 9 different pipelines for the generation and benchmarking of the CRISPR gene knockout consensus signatures. Each pipeline consists of one of 3 consensus approaches and one of 3 similarity metrics, as shown in the slide. Using the values derived from each of these pipelines, I paired each of the knockout genes with the corresponding gene that had the highest correlation value. After assigning pairings for each knockout gene, I then kept only the top 100,000 gene pairs. This list of top gene pairs is what will be benchmarked against BioGRID's PPI database. For benchmarking, I utilized the top 150,000 associations from the BioGRID protein-protein interaction. Earl Evangelista from the Mayan lab extracted this list of PPIs from the BioGRID download file repository for another project, and I was able to use this data for this project. Only human-human interactions, physical interactions, and direct associations were taken into account, and the top 150,000 interactions were then ranked based on confidence values. When first organizing the PPI dataset to be used for my benchmark, it was observed that only 3,831 genes overlapped with both the PPI database and the original knockout genes. This was taken into consideration by evaluating just the overlapping genes when benchmarking. The top 100,000 gene pairs from each pipeline that overlap with the PPIs in the BioGRID database were used as the benchmark. A randomized method in finding these gene pairs was included as a control and provided an additional form of comparison. Specifically, a shuffling method was applied to each pipeline in which the rankings of the gene pairs from each pipeline were shuffled, in addition to a generalized randomized method that entailed randomly pairing 2 unique genes from the overall list of knockout genes. The bars for these 2 methods represent the average from shuffling gene pairs 100 times and recording how many genes overlapped, and the error bars of these methods represent the standard deviation from this process. The gene pair rankings obtained from 9 pipelines had significantly more overlapping genes between the PPI database than the randomized and shuffled methods. This indicates that the pipelines were effective in finding high correlations between genes and thus generated valid consensus signatures. The pipelines that utilized the highest effect method were not optimal in generating consensus signatures. The pipelines that gave us the highest overlapping genes were both the Pearson mean and cosine mean pipeline with 200 overlapping genes. To further visualize these results, 3 pipeline-based bridge plots were generated to see if the pairs from the PPIs are highly ranked within the most correlated gene pairs from the CRISPR knockout data. These bridge plots validate that the pipelines that utilize the highest effect method were the least effective in generating high-quality consensus signatures, while the median pipelines worked slightly better than the mean. To conclude, the L1000 consensus benchmarking project formulated 9 pipelines to produce consensus signatures from 140,945 total signatures. 9 different pipelines were benchmarked with the BioGRID PPI database. Further directions for this project include generating more consensus signatures, generation methods, and applying a similar benchmarking process to the chemical perturbation signatures. The creation of drug-drug similarity matrices from other sources, such as chemical similarity, could be used to benchmark. I would like to thank the entire Mayan lab, including the summer trainee cohort, for their help and advice throughout the course of the summer. I especially would like to thank Sherry Shea for answering my questions and Earl Evangelista for providing me with a PPI database. Finally, I would like to thank Professor Mayan for the opportunity to work in the lab and for his continued support and patience. Thank you for listening.